We're live. Okay, so hello. I'm Cristina Peristeridu. I'm an assistant professor at the Faculty of Law and a member of the Executive Board of FEM. I would like to welcome you all to the first Women Researchers Festival of Maastricht University that FEM co-hosts with Pint of Science. As you may know, FEM is a network of staff at Maastricht University with a mission to promote the empowerment of women and female leadership at the university and to foster a culture of gender equity and diversity. The event is to celebrate the innovation, curiosity, and daring nature of women researchers at UM and honor the International Women's Day, which is today. We would like to provide a platform for women researchers to share their research and perhaps to foster collaboration. We have been more than happy to set this up together with Dr. Anna Soet from FPN and the Pint of Science, which dedicates its work to make research open and accessible. So today we will hear the presentations from 16 researchers divided into 2 sessions. So this session has 8 researchers. I must say that we received quite the amount of applications, so we had to make a hard choice. Those not chosen to present have been offered the opportunity to publish a research poster, which will be shown at the website of Pint of Science later today. So with no further ado, I would like to give the floor to the researchers. Each presentation is about 5 minutes, and then we will get about 5 minutes for answering questions. And also, I must say that our event is recorded and will be shared later on social platforms. Okay, so let's begin with our first speaker, which is Dr. Nicole Basaraba. She's a postdoctoral researcher at FSE, and she will talk to us about storytelling. Nicole, you have the floor.
Thank you. I will put it full present mode so I can see nobody. So someone tell me audio if something happens with the presentation. So thank you for having me. I'm brand new to Maastricht University. I've been here just over 1 month now, and today I'd like to share with you the direction of my postdoctoral research for the next few years. So that is on the topic of digital storytelling and European heritage. So the research that I'm looking at specifically now is this concept of digital placemaking. So the word itself you may not have heard before, but you might have experienced this either using a mobile app to, go on a walking trail for heritage, or maybe you've been to a museum or you've interacted with a digital display looking at archives. And here you can see a picture from the Studio Europa, which is what I'm part of. And they've created this interactive website where you can click on these gray dots there, and these pop-ups will come up, and you can explore the history and culture of Maastricht City. So the aspects that I'm interested in is this location-based aspect of digital storytelling, because it's growing in the number of disciplines who are examining it. So it actually started in geography and urban studies, and now it's actually merged into the creative arts. And so that's kind of where my research comes in, is looking at these aspects all put together. So the main research question that I'm looking at is how these digital placemaking techniques can be used to create stories of shared heritage in the Meuse-Rhine region, which is pictured there in this map. So the idea is that engaging the public with this history, so not only we're creating digital stories just to put something out there, we actually want to hear from the public and democratize the knowledge building process. Because nowadays we have all this digital technology, audiences are not just passively consuming knowledge that are produced by historians and other experts, but they're also creating material themselves. So they're creating, Twitter posts and blog posts and engaging in genealogy research. And historians and other experts have started to consider how this user-generated content can be given a voice and incorporated into shared authority with heritage. So the sub-themes that I'm investigating under this umbrella of storytelling for Europe is this idea of cultural identity And I'm specifically interested in how this can be defined with cities that have a history of changing borders, especially in this region. So for example, in the Netherlands, Belgium, and Germany, some of these borders have shifted in recent times in the scale of European history. So how has that affected their local identity as well as their European identity? And I'm interested in what their common layers of shared heritage are at specific locations. So again, this brings the idea of place and how we can use digital technology to tell the stories of these shared places with cultural heritage. And then the last aspect I'm looking at is how citizens can participate in this heritage making and also how they respond to digital placemaking practices. So these kind of interactive websites and mobile apps, how do they respond to these stories of heritage? So the question is how I'm going to investigate these rather complex and heavy questions. I'm going to build in citizen science or participatory methods into my research. So in the UK and Ireland, this is called engaged research, and it also draws from that discipline of public history. So the main way I'm going to do this is through many case studies. So I'm going to do case studies on border regions, as well as looking at examples of capitals of European culture, because they are portraying their culture and their heritage in a specific way, and they're determining how they do that. So I want to see what are the patterns, and I'll do this using what's called big data analysis. So I use computational methods to look at, for example, the Europeana archives and social media data, and I analyze this and use topic modeling to let the data and the existing communications of heritage about Europe and specific cities speak for itself, and then I can kind of put this back together and see how it informs European identity and heritage. So the potential research impacts for this work is that I plan to advance this idea of creative digital placemaking, which is actually a rather new concept in the literature because it is now expanding to multiple disciplines, especially in the past 5 years. And then it also has an economic potential because now we're living in a COVID pandemic. There's virtual and local heritage tourism options with this kind of digital placemaking. And then finally, it looks at how citizens are receiving their shared cultural heritage and identity. What are they saying about it rather than, you know, the academics coming in and telling them what they think? It's vice versa. It's a bottom-up approach. And then finally, what's next in the creative industries? And that comes to this newness of this digital placemaking in terms of storytelling. So that is my 5 minutes, and I'm looking forward to hearing any questions you have about my unfolding research and hope to connect with you further.
Okay, thank you for this most interesting research and very also relevant for this region. Huh?
Yeah.
Okay, so let's see whether any questions will pop up. It might take a while.
From the first one too, so people are having their moment to get settled.
Yeah. Okay.
So,
you can read it, Nicole, right?
Yeah. So thank you, Linda. What are the major challenges in digital storytelling? What are the success factors? That's a very good question. I think the challenge with digital storytelling in a heritage context is that it is something new, and we have all of these new types of digital technologies like virtual reality and augmented reality, like the Pokemon Go, where, you know, the thing jumps off the screen at you. And we have all these new technologies, but no one's quite sure how to use them in a strategic way to engage with the public. So I think strategy behind using digital storytelling is a big challenge. And success factor, I think, is determined by who's using digital storytelling. So for example, if it's a museum, if they're going to engage in digital storytelling, the question goes back to them is, why are they doing it? What do they want from the public? Do they want crowdsourcing? Do they want engagement? Do they want to share an untold story? So I think the success factors are determined by the creators, and then they can develop metrics on how to measure their success. So what is successful engagement for them? Is it a number, or is it awareness, and these kind of things? Hope that answers your question. Thank you.
Okay. Oh, another one.
Thank you, Sharon. Are there any potential social impacts to your research, or that is not in your scope? So yes, in terms of social impacts, I'm hoping that there would be, in terms of connecting with citizens and letting their voice be heard. So my research, I like to focus on these untold stories and the alternative perspectives. So that's why in my case studies and my big data analysis, I'll be using content that's actually user generated or produced by the public, so that they are included into the story. And if we are, again, doing digital storytelling for them, maybe they learn more about their history, their heritage, and their culture. So it could be beneficial for them, especially even for children, for students as well, like to engage with these platforms and learn about history and heritage in an interactive way. Thanks, Sharon.
Okay, let's see whether there is any more questions. Okay.
So thank you, a great presentation. I was curious to know how this could be translated across different cultural boundaries. Yes, so that's one of the aspects I will be looking at, and that's why I'm interested in these border regions, is to investigate, for example, language, how language plays into it, and also this idea of nation versus collective. So, so since my focus is on Europe, I would be using European case studies, but again, this can be expanded to different cultures. So that's why we're lucky to be academics, and that we can work with theory, and then this can be applied to different case studies. So my research is in media theory and media studies. And so the results of my research would intend to be applicable and taken upon by other creative agencies in different countries and different nations. So my goal would be that it can be translatable to different cultural boundaries outside of Europe as well.
Okay, so thank you very much, Nicole. Let's move to the next researcher. We have Dr. Natalie Weiss. She's assistant professor at FHML, and she will discuss today her research about gut-brain interactions, which sounds fantastic, no? Only the name.
Yes. Thank you. Thank you. So, have you ever felt your happy or sad feelings with your gut? Or felt your anxiety or excitement with some kind of a stomachache? Or even butterflies when you fell in love? Well, you're completely right. And that's because our gut harbors our second brain. So, as mentioned, my name is Natalie Vaas, and I'm a postdoc in the group of Dr. Velemelot, and I would like to tell you a little more about the gut-brain axis and give you some more information about our brain, and especially about our second brain. Let's see if I can move forward. Yeah. So our brain consists of about 170 billions of neurons and glial cells, and together with the spinal cord, it forms our central nervous system. And the central nervous system communicates with and is connected to all other organs via these yellow nerve fibers you see here, the so-called peripheral nervous system. An important and almost completely autonomous part of this peripheral nervous system is the enteric nervous system, or so-called second brain. And the second brain consists of about 200 millions of neurons and glial cells and is located along the gut wall through the entire length of our gastrointestinal tracts. And in total, this enteric nervous system is about the size of a cat's brain. Although the cell types in our first brain, so the one in our skull, and the second brain are pretty similar, there is one main difference, and that is that the neuronal layers in our brain are quite structured, whereas the neurons are rather randomly distributed within the enteric nervous system in our gut. So here in this schematic figure, you can see that the neurons are depicted in red and the glial cells are depicted in blue, and they're located here in the gut wall in between the muscle layers. And the enteric nervous is pretty important because it serves to regulate all of our intestinal functions, like the motility of our gut, and it makes sure that we digest and absorb the nutrients that we need every day. And this is to make sure that our intestinal homeostasis is maintained. So proper functioning of this enteric nervous system is essential for life. And it is therefore involved, or it can be involved in several intestinal diseases like inflammation or Hirschsprung's disease, but it is also involved in diseases of the 21st century via the so-called gut-brain axis. So there is an involvement in, for example, Alzheimer's disease. The role in colorectal cancer is, however, still unknown. And this actually came across our path because my supervisor, Dr. Malot, identified that NDRG4 is an important biomarker for colorectal cancer, and I found out that NDRG4 is specifically expressed within this enteric nervous system. So this you can see here. This is a picture of our gut, and you can clearly see this brown staining within the enteric nervous system. And more specifically, we found that NDRG4 is specifically located in the enteric neurons and not in glia cells, because you can clearly see an overlap between this red neuronal marker and the green NDRG4 fluorescence. Using mice that were deficient for NDRG4, so mice that didn't have NDRG4, we saw that they had enlarged intestinal tumors that were of a more aggressive phenotype, as depicted here above in these pictures. Also in vitro cell cultures, we found that if you boost intestinal organoids with the medium that was derived from this enteric nervous system without the NDRG4, we also saw that these organoids start to grow faster. And then we used mass spectrometry to identify the factors that were responsible for this mechanism. So we found out that these enteric neurons without NDRG4 start to produce higher levels of two extracellular matrix proteins, named nitogen-1 and fibrillin-2. So we thought that this suggested that upon loss of NDRG4, your enteric neurons start to produce and secrete more of this nitogen and fibrillin, thereby stimulating the growth of intestinal tumors. To date, I am further pursuing this research line with my Coach Talent Fellowship grant, and I'm trying to figure out whether this communication between the ENS and the epithelial layer, so the layer where the tumor starts to grow from, is direct, so direct signaling mechanism, or is either indirect. So maybe the ENS first signals towards some sort of an intermediate cell, for example, an immune cell or a macrophage, and by this way, then this immune cell communicates towards intestinal epithelium, thereby stimulating the growth of these tumor cells. For this purpose, I'm using a zebrafish model because the zebrafish are transparent, as you can see in these pictures, in their early life stages. So it means that you can easily follow fluorescently labeled cells or cellular components over time. And here I show you an example of a zebrafish that has fluorescently labeled enteric nervous system, and on the right you see a zebrafish that has fluorescently labeled neurons in green, and I don't know if you can clearly see it, but in red there are also fluorescently labeled macrophages to further investigate our hypothesis. Now, of course, I want to acknowledge the groups that I've been working in, and this is my group in Maastricht and also in Rotterdam, and especially my supervisors, Dr. Malot and Professor Hofstra, who have been guiding me throughout this project. And as you can clearly see from these pictures, both of these research groups already have a lot of women in there, so I believe that our groups are on the right path, empowering women in science. So thank you all for listening, and I'll be happy to answer any of your questions.
Okay, thank you so much for this amazing topic. Very interesting, and especially the zebrafish. Okay, so let's see what questions will pop up on the screen. It always takes a minute. Oh, okay, from Anna.
Okay, so thank you, Anna. What type of a microscope did you use? For the zebrafish, we are using a confocal microscopy, so that we, of course, can really zoom into really deep into the different layers of the zebrafish, because we want to image the different, like an embryo, like a zebrafish embryo. So we really want to go deep into the zebrafish layers. And for the other fluorescent images, we also use the confocal microscope so that we could clearly see in the ENS where that the cells were located. Thank you, Anna.
Okay, thank you. Anna Soot makes amazing microscopes. Maybe you can use hers. Okay, let's see another question. Okay, there it goes.
Okay, thank you, Tachi. So there is indeed a link between gut flora and mental health disorders. And we can also investigate this in zebrafish, but we have to take into account that the gut flora in zebrafish is a little different than that we have in mammals and humans and in mice, because of course these animals live in water. But I believe that about 80 to 90% of the gut flora is similar in zebrafish. So that is also indeed a link that can be pursued using the zebrafish model.
Okay, thank you. Good point. Okay, another one.
Okay, Linda, are there specific foods that keep your second brain happy, like chocolate for the brain? Well, I think that chocolate for my gut is also very good for my mental health. So, but that's probably not the best choice for your gut, but food that contain a lot of fibers and the healthy food, of course, will keep your second brain happy. And if you go too much to the McDonald's, I don't think your second brain will give. They will give you happy signals for an instant, but afterwards, that happy feeling will quickly fade away.
So that's a reason for all of us to get probiotics.
Maybe.
Okay, let's see whether another question will pop up. Maybe not. Okay, then maybe, yes, maybe we can wrap this up. Thank you so much for this most illuminating, like the zebrafish presentation. I think it's a lovely topic, very interesting.
Thank you
so much. Okay, so next is Dr. Jeanine Johnson. She's actually a doctor, a medical doctor and researcher at the academic hospital in Maastricht, the UMC. And she will present her research on sacral neuromodulation in women. I hope I pronounced this correctly. So you have the floor, Jeanine.
Good morning. My name is indeed Jeanine Johnson, and I'm a clinical researcher at the urology department. And I'm going to present you a study design where we are looking at the influence of sacral neuromodulation on brain activity during bladder filling. And I want to start by presenting you a case of a woman. This is a case that we see very often. It's a woman 50 years old who comes with complaints of urgency and frequency. She tells us she has to go to the toilet at least 20 times a day. She has to rush and often loses urine because she still doesn't make it. And these complaints actually exist for a long time, like 8 years. In that time, she's seen multiple doctors. She tried a lot of other treatments, physical therapy, medication, but they're all not successful. And as you can see here, overactive bladder is a syndrome of symptoms. It has a very large disease burden because these women, sometimes also men, but mostly women, they are often very young and they don't go out of the house without knowing where the toilets are, and they're always afraid that other people see their urine loss, and so they stay at home a lot of the time. So it affects their life greatly. The prevalence is also a lot larger than you would think. It's around 10% in middle age, and it only increases with age. And what you see here in this case is a therapy-resistant overactive bladder, and that's what we see a lot here in Maastricht. And these people have an indication for sacral neuromodulation. Sacral neuromodulation is a third-line minimal invasive treatment. At this center, it has a success rate of 76% in patients with overactive bladder. But deciding which patients are eligible for this treatment and which are not is very difficult, because we don't know at what kind of problems we should look. We know that people with overactive bladder they have an indication, but that's all we know. So we do this surgery in 2 stages. As you can see here, you have let me see, you have this lead that goes to the sacral foramina. That's what we implant in the first stage. And then when the treatment is successful, we also implant a battery, which is very expensive. We know that the sacral neuromodulation system gives an electrical stimulation to the nerves that regulate bladder activity. But the exact working mechanism is not known. It is believed that the neuromodulation system modulates the spinal cord reflexes and the brain networks by stimulating the peripheral sensory and possibly the motor neurons. But it has been very difficult to test this because up until last year, it was not safe to put patients with a neuromodulation device into the MRI. But since last year, we have an MRI-compatible system, so now it is possible. In our study design, we use a pre-test, post-test group design where we include all female patients with overactive bladder and an indication for sacral neuromodulation that present themselves at our center. And of course, they should have no contraindications for MRI. We do 3 measurements at the same time. We put the patients underneath a 7 Tesla MRI scan, and we fill the bladder while recording urodynamics, which you can see at this image. And next to that, we let people fill in a bladder sensation scoring system, which you can see here. So they can tell us how full they think their bladder is. We are going to measure this at 3 different points in time. We will start at a baseline where people don't have a sacral neuromodulation system yet. After this first scan, they will get the first stage implantation, which you can see here in the images. The lead is placed and stimulation is started. Then in the testing phase, which lasts for 2 weeks, we do a second MRI scan and we see what the short-term differences are. And if the people are successful, we do the second stage implantation. So we place the battery. After that, we do a long-term follow-up scan, and that's after 2 to 3 months. And we also look at brain activation during bladder filling. What we, of course, want to know is what the influence is of the sacral neuromodulation system on brain activation during bladder filling. We calculated the sample size by looking at other articles where they tested the same. And we need 15 successful patients, because only in successful patients you can do the long-term follow-up scan. And looking at the success rate that we have here in this center, we need to include 20 patients. And our hypothesis is that we will see short-term changes because we also see a quick clinical effect in people. And that we will also see additional long-term changes because of neuroplasticity. So we expect a difference in long-term and short-term. Are there any questions?
Okay, thank you for this lovely presentation. Let's see if there are any questions. It should pop up any moment now. There it is.
Okay. Could sacral remodulation help battle the symptoms of kidney stone disease and the prolonged use of stents? No, we don't know that because the kidney stones are formed in a different way. They are formed because of how do you say it? Differences in the concentration of different different minerals in the urine, and the sacral neuromodulation system doesn't help with that.
Okay, but like, because now another question from Teresa Schumann.
Yes, thank you, Teresa. What I was wondering was at what areas you look in the brain. Would non-invasive brain stimulation also be an option eventually? We look at mostly at the PAG, That's because that's a connection center which leads the stimulus to the rest of the brain. And there's actually a colleague of mine who is now busy with research here, looking at the PHE with the 7 Tesla fMRI scan. They see that there's a difference between people with overactive bladder and people without any bladder complaints. So then we can use the results of that study to also see the differences when we use sacral neuromodulation. And if non-invasive brain stimulation would be an option, for now we don't know that that has an influence on bladder complaints. So who knows? But for now, we don't use that at all for these kind of complaints.
Okay. Oh, another one.
Another one. Thank you, Sangeeta. What would be the result of the neuromodulation? What would be a good indicator? What we do is we look at the results by letting people fill out mid duration diary. And we think that the neuromodulation system has a good result when we see a 50% decrease of the symptoms in the patient. But we don't only look at the numbers, but we also look at how the patient feels and if they think it has an improvement in their lives. So that's what we use to decide if it's successful or not.
Okay, thank you. I was wondering, I had a question myself. Could you use the results of this research for incontinence that many older women suffer from?
Yes, because we see that urgency incontinence, which is when you need to pee and you don't make it to the toilet, has a very good reaction on sacral neuromodulation. So that definitely can help these women. But there are different types of incontinence, like you also have stress incontinence, which occurs when people have to cough or sneeze, and that cannot be treated with the sacral neuromodulation system. So it depends what the cause is.
Okay. 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 So we need to move forward. If there are any more questions, we can pick them up afterwards. Okay. So thank you so much for this presentation. Now let's move to our next researcher, Dr. Dorit Bierman, who is a postdoctoral researcher at FHML. And her presentation is about health in action. What do you mean by that, Dorit? Oh, we cannot hear you. I think now, can you speak maybe now?
Yes, I think now it's...
Yes, it's great.
You can hear me. Okay. Yeah, I only have one monitor, so I have to juggle. Yeah, my name is Dorit Bierman. Thank you for introducing me. I'm a postdoc researcher on the research project Health in Action, and I will tell you all about it now. The main focus of this research project is workplace health promotion. And I do this together with my colleague Lotte Thyssen, Agnes Meersuk, and Klasine Horstmann. We also have a website if you want to have a look. Why we do this research. Every organization strives for a healthy and corporate culture, and that with people who are vital, resilient, and of course productive. However, many organizations struggle to get employees involved in health promotion activities at the work floor. And therefore, this research project has the aim to create workplace health initiatives that are attractive to employees and also sustainable. And the approach we use is an ethnographic participatory approach that puts the perspective of all employees central. Ethnography is a method originally from cultural anthropology, and by means of fieldwork, the ethnographer actually tries to understand from within what is happening and what is normal within a certain community or group, so how things are done around there. Ethnographic research has also been used in market research to investigate user experiences. For example, Lego has done ethnographic studies around the world to see how kids are playing. And the customer is put central here. Ethnography is a really valuable research method to get deeper insights and often questionnaires do not offer the space to say why or you do things or you don't do things or how you feel about it. And this example illustrates this a little bit. How likely are you to recommend Windows 10 to a friend or colleague? And the respondent answers not at all likely. And he explains, I need you to understand that people don't have conversations where they randomly recommend operating system to one another. So it's actually learning by participating. And Lotte and I worked along in organization. We lunched with employees in the organizational context and had a lot of informal conversations and also interviews in order to understand the living worlds of employees and their understanding of health. So what employees actually need in order to feel healthy. And the ethnographic method enabled us to get deeper insights into the perspectives of employees. And I selected 2 quotes here to illustrate that for employees, health at work is more than lifestyle, for example. You need more than fruit alone. That's not what people need. Better yet, you need to start on the other side, that people feel part of the company and that they find it normal to participate in activities. Someone else said, he has not worked a night shift for ages. He's talking about a colleague. Because of his migraine. The other shift members took over his night shift, and that is flexible and matey. So these quotes show that health is about belonging and also caring for one another and about social relations. Another frequently topic was the qualities of a good team leader. Like the following quotes illustrate, this was a team leader who said, I don't think the board knows how important the knowledge is that I have about each employee. I know how to play the game every day, but you don't know these things just like that. That is the result of year-long investment. And employees also said about the team leader, which they valued, he expresses appreciation. He treats cake every now and then, and it's that little bit of extra attention. So we supported organizations in setting up health activities with employees, and activities ranged from workshops about healthy food, line dancing, and also, for example, drumming. Another organization, for example, organized it among other activities, gymnastics on the work floor, and through this ethnographic method, we could grasp that an organization is more than this. So an organization and its structures are characterized by its history, its social relations, and what it matters most, their people. So mapping the organizational context and also the perception of employees, what they regard as healthy, is really important. And to conclude from this, it's when people feel heard, they can better identify themselves with the organization, and organizing activities together with employees creates more commitment, ownership, and connection. And this has a positive effect on employees' well-being and also promotes participation in health activities. Thank you for your attention, and I'm happy to ask questions. I have to answer any questions.
Thank you so much, Dorit. So let's see whether there are any what questions you will get. I mean, I already have one, but I think I will wait to see what the attendees will like to ask you. Maybe in the meantime, I can ask my questions. Okay. So what would you, bearing all this in mind, what would you suggest as an organization to do based on your research?
Listen to employees.
Okay.
Yeah.
Listen to this, but maybe also because you mentioned that you would like to have more that it would be good to have collaborative events where people gather and do like drums or.
Yeah.
Would this be something that you would suggest, or in a structural manner, like once per year, a faculty can go do silly things together?
Oh, I think, of course, you should first ask people and then organize it with them, and then you have also the commitment, because it's that top-down strategies that are really sometimes, people don't, feel welcomed or don't feel that it suits their mindset. So it's actually about listening what people are in need or what they think, and then together with them organize activities, and then you have that commitment what I was talking about.
Okay, so that's very okay. That clarifies a lot. So it's really the bottom-up approach that makes a difference.
Yes.
Great.
Because if I organize, sorry.
How similar are work-life balance and health?
How similar? Yeah, I think it's a big. I mean, work-life balance. Then you kind of separate them, and I often see or think that you can't separate work from life and life from work, because it's entangled. And I think also with the home office situation now, we see that even more. Work is in your home, and then balancing it, it means that they are two separate things. But isn't work part of life also? We could ask ourselves. I don't know if that answered your questions. In your research, you do pay attention to the able-bodied, disabled divide. That's an interesting question. I think it's also about everybody does what he can do, and not maybe you can specify your question a little bit more, because I think maybe I head in the wrong direction.
I think she means whether the definition of health, or how health in research have like an element of diversity in terms of what people, in terms of disability.
Of course, I think for everyone, health is very, very different, and also that changes over the life course as well.
Yeah. Okay. Oh, another one. Do you see any difference between participants from different countries near the Maastricht area?
We actually only did the research in the Netherlands, but as I am German, I would say that there is not a very difference in the countries, but it's also in the organizations. They are very different. So I think every organization has a different culture, if you could say it, and also that also influences how participants perceive health, or perceive work, or perceive their work-life balance. You could say that.
Okay. Shall we wrap it up, or is there any more question coming? Let me see. I think this is it. Okay. So thank you so much for Dorit for this very interesting presentation. I hope that you have picked up on it, and we will see some results from it. Okay, so let's move on to the next researcher. I would like to welcome Dr. Therese Gronert. She's an assistant professor at SBE, and she will discuss something really important: overcoming hierarchy barriers. We need that.
Absolutely. Thank you very much for the kind introduction. If everything goes well, you can see my slides now. Indeed, I study something that we all do every day if things are going well, even though we may not notice it. I study how we as professionals develop knowledge when we're faced with challenges or novel problems. So how we grow professionally and how we develop ourselves. This is something that can take place simply by observing how a more experienced colleague is doing something, asking questions, and also having difficult conversations with our colleagues.
as you can see from my title slide, I'm particularly interested in when this kind of learning does not take place as it should, the barriers towards learning. And one of the barriers that I focused on in the last few years, together with my colleagues Roger Meurissen and Wim Gijselaars, is the barrier that we experience when we are in a hierarchical context. We all intuitively understand what a hierarchy is. For example, think of your direct supervisor at work or of your youngest sibling, and you know exactly what I'm talking about. So in the past few years, we have explored how leaders and followers experience the process of learning from each other when they work in a team. And we've explored this in many different ways. We've conducted interviews, experiments, and surveys in a number of accounting firms, which is a very hierarchical business environment, even worse than university by Dutch standards. Now, the first thing we found, which isn't really that surprising, is that leaders and followers have the same concerns when it comes to learning from each other. They essentially share a similar fear, and that is, what if they think I'm stupid? As a follower, you may remember this very well, your first few days of working, you wanted to make a good impression on your leader, of course. So maybe you were hesitant about asking questions, you didn't dare to point out problems, or even that you didn't disagree with something, even though you knew you had a good point. The leader, on the other hand, is not immune from the same problem either. They also want to be seen favorably by the people that they guide. They want to have authority. And for the leaders, this resulted in behaviors such as not sharing uncertainties about the project that the team was working on, not being willing to admit mistakes, which is a big one, and not daring to say, I don't know. And in teams where we found this kind of dynamic, where people are more concerned about their image than really about developing and doing the best that they can, learning is very much inhibited. Fortunately, not all of the participants in our studies showed this kind of thinking pattern. We also had a few people who approached teamwork quite differently and in a much more productive way. So, focusing on leaders first, we had several who, instead of asking, what if they think I'm stupid, they asked, what can I learn from them? And they particularly valued the new insights, innovative ideas, and fresh perspectives that younger colleagues can bring to the team. However, for the team to benefit from these ideas and to exchange these views, they had to show specific behaviors that facilitate this kind of exchange and learn. So if you are in any kind of leader position, formal or informal, take note of the following behaviors to get the best out of your colleagues. First, show personal vulnerability. If you're not sure, let your colleagues know. It's human, it's fine, and it normalizes doing the same for them. Share your own mistakes, and in fixing the mistake, invite your younger colleagues to participate with you so that you show them how things work, but also you value them and their professional knowledge. Seek feedback, whether or not you're creating an environment that is effective for them to learn with you. In short, role model psychological safety. However, there's a small caveat. We found some different patterns for female leaders and female followers. It appears that the female professionals that we studied were much more sensitive to the display of these behaviors in terms of how consistently they were shown and how frequently they were shown. Male colleagues thrived on fewer of the behaviors. Female professionals only engaged in learning when those behaviors were shown very consistently across different leaders. The second thing, and please ask me about this in about 2 minutes' time, we found that female professionals needed to prove themselves significantly to more experienced male colleagues. And I'm very happy to share more about that in the questions. Now, we know what a leader can do to facilitate learning. What about the followers? What about the younger colleagues, those that are more vulnerable in the hierarchy? Here we found that followers asked themselves, what can I learn from my leader, when the leader had role models, learning, and psychological safety. Then they really benefited from gaining high-level insights, personal experience, as well as more contextualized knowledge from their leader. For example, if A happens, B usually works well, things that take us a while to pick up. In those moments, the followers were more active in asking for information, help, or feedback. And specifically in the audit setting, what was very effective was inviting younger colleagues to join in a task that they were not quite ready for yet, and then to debrief and reflect together how that task works, to prepare the younger colleagues to slowly gain the knowledge they need to rise through the hierarchy. Now, if you are a leader, you may think this is a lot of pressure, right? You are not the only one that matters, though. There are some contextual factors that help you out. For example, if you have the time to build trust and familiarity with your team members, that's fantastic. If you have a shared goal in your team you want to achieve, even better. And a little bit ironically, we found that spending time face-to-face is a very important factor. So very critical in these times to find a way to compensate for that. Now, this is, in short, the research that we've done in my team, and I hope that I can tell you more. Thank you very much.
Wow, Therese, what can I say? I mean, lovely research, very interesting results. I have already dozens of questions myself. First of all, I think at FEM we should really invite you to give a presentation at a lunch meeting, because I find your research extremely relevant and important. Let me see whether there's any question coming from the audience. Yes. Okay.
Yeah, so looking at different kinds of organizations, we have focused our research on audit firms. So it's one kind of business setting, but they have different sized organizations in there. So we have the larger 4 firms and we have smaller companies. We found that in smaller firms, the hierarchies were flatter because people work together more frequently, they spend more time together, and there are fewer hierarchical levels within a team. So that can be a benefit. However, the larger companies had a larger network, so people got to know more people. They had an easier time finding coaches outside of their own teams. So there's pros and cons, but I can't speak to startups, for example, or to smaller companies.
Okay, thank you. Until the next question pops up, just let me ask you, what is psychological safety?
Psychological safety is the feeling that you can ask questions or disagree with someone without being thought of as mean, incompetent, stupid, or worth less than others. So it can start by things like, this may be a stupid question, but are we going in the right direction? Then people can either react like, oh, she doesn't know directions, or they can say, let us check. Right? So it's in the small, simple behaviors that show appreciation for each other, for someone taking a personal risk to help the whole team perform better.
Okay, that's very interesting. Okay, let's see whether there are any other questions. Oh, how can we put it? Okay.
Yes, so how can we break down hierarchical barriers, high context and low context cultures? So I've conducted this work in the Netherlands. However, I'm German myself, so I already have some minor differences in how hierarchy can be experienced. The important thing we found is that leader behaviors are determining how hierarchy is being experienced. So if I use someone's last name, if I don't invite people for small questions, small interactions, if I make short statements and then I leave, that is a signal that I'm above you and that I don't invite you to come up to my level. If I show personal interest in you, if I ask you what you need to learn and develop, if I invite your opinion, I'm showing that I value you as a person, even though formally we might have different levels in an organization. The behaviors that are needed to make this difference clear are very likely to differ drastically across cultures. In the German work experience that I've had, the leaders showed their appreciation differently than they do in the Dutch culture, where we say, we use first names for everyone. You're very free to disagree with everything and everyone all the time, and no one thinks very badly of it. That's not the case in Germany necessarily. So it's about valuing someone as a person with their knowledge, with their experiences and their perspective, regardless of where you stand on a formal rank. The behaviors that determine that will differ across cultures. And we definitely need more research on this.
Oh, very relevant question. Okay.
Absolutely.
Yeah, thank you, Teresa, so much for this lovely presentation. We will be in touch with you. Anyway, okay, so thank you. And let's move on to, Dr. Katerina Stankova, who is an associate professor at FSE. And she will discuss dynamic game theory. Oh.
Okay, first of all, thank you for inviting me to this really interesting event. And of course, I am really sort of intrigued by the talks which we hear so far. And I come from quite a different background. So I am an applied mathematician and game theoretician by myself. And I coordinate dynamic game theory at the Department of Data Science and Knowledge Engineering at the Faculty of Science and Engineering at Maastricht University. So I'm going to talk a little bit about mathematics, not too much, mostly about the concepts. So, and of course I have 5 minutes, so I will try to not spend them by mathematics only. What is game theory? Game theory is mathematical theory of decision making. And in fact, every situation in which multiple parties interact and where one's outcome is actually dependent not only on their own actions, but also on the actions of others, like, for example, this event, is a game, okay? And then the team that I coordinate, the dynamic game theory team, focuses on all kinds of games, so those which evolve in time, those which actually have some unknown elements, etc. Basically, we are mostly interested in dynamic games because those are situations which actually evolve in time, like, for example, this event. Okay. Basically, we are sort of interested in expanding the theory, but also in all kinds of applications of this theory. So often we are interested in if I'm rational and I'm included to some problem, can I solve them? Okay, so some of them are like very classical ones. In traffic control, if you have a lot of congestions, can you actually do something smart? Like include traffic lights or include maybe tolling of the roads to actually somehow influence the drivers, which might be a bit selfish on their own, to do what you want. Here, for example, to minimize congestions. Or maybe how to be a little bit more smart with our energy. Or maybe how do you make your employee to do a little bit more effective work, although they are not really interested in it. And then we also focus on like environmental problems and also on problems which involve biological resources. So for example, here, the idea is, okay, if I fish too much, the fish is going to respond by actually shrinking its size. So we use game theory to actually explain why it's happening and also what we should do different if we want to actually keep our biological resources. And also if you are a fisher, to actually maximize your profit, because fishing as much as possible really leads to shrinking of the fish. So this is not going to help you much. And the most passionate application I'm the most passionate about recently is cancer treatment. So we are applying game theory to actually explain and optimize cancer treatment. So I think I'm a bit slower than I thought. So, but we are also interested in kind of classical game theory. We are interested in the situation of rational players interacting. Then in evolutionary game theory, we are interested in more Darwinian evolution. So how do our players actually evolve given the conditions to whatever is happening to them? And we are sort of trying to expand models of both these types of games, but we also try to look on a connection between them. So those are examples maybe of the applications which I was showing before. So if I have an evolutionary system like fish, like cancer cells, like pests interacting with on different plants, if I now introduce to them actually a rational leader, can this leader drive the system into something what we would want to have it in? Yeah. If I repeat this, my main question is, if a rational leader enters a dynamic game, can she drive the system into a desired state? And so I was already mentioning a bit cancer. So what we are actually achieving by our game theoretical model is to change the standard of care in metastatic cancers. Those are cancers which actually already left their initial site and they are spread all over the body. And by using our modeling, we actually could show that the standard of care often leads to tumor progression, while actually more dynamic protocols of treatment could actually keep the tumor under control. So here you see how big the tumor is. And with our more dynamic protocols, we can keep it under check. I don't believe I can explain all of it in the remaining 30 seconds, but I'm, of course, very interested in taking questions and I can explain it further. And then basically, of course, for our models, we need real data. So this is a commercial for the next speaker, Veronica, who is going to actually talk about how can we use, for example, genomics data to feed our game theoretical models. Okay, that's it for now. Thank you for your attention. And I would like to thank the Dynamic Game Theory team. And if you have Twitter, please follow the team, follow me if you like.
Okay, thank you so much. Wow, what a topic. So let me see whether there are any questions. I see one actually. Let me see whether this can appear on the screen. Okay.
So the dynamic game theory being used to model the global reaction towards good. Yes, this is a very good question. Indeed, so there exist already game theoretical models, and we have been playing with this topic as well, where we actually really sort of, this is a typical example where you could show that if the leader is rational and sort of can foresee their action, they can do much more than if you are just reacting to what's going to happen. So that's clearly, so you see in many countries, they just sort of watch what's going to happen and then they do something, yeah? And if they would already in advance somehow consider like, okay, there will be these new strains appearing, if we sort of, target just this one, they would do something slightly different, right? So you see that if you learn from your past mistakes, you can do much better. So indeed, this is one topic which we are also looking into. Okay, and there is another question. I'm not sure if, how do these models work in practice? And how effective are they in comparison to current models? Are you now, I'm not sure if you're now asking about cancer. So in cancer, we are basically having a collaboration with the Moffitt Cancer Center, who is like leader on these kind of therapies, which we call evolutionary therapies, which are cancer therapies, which actually anticipate and steer evolution in cancer cells in response to treatment. And there we are actually testing the clinical, the strategies which we develop in clinical trials. And from the results of these trials, this looks very good. So they sort of, first of all, use much less drug, and you keep these tumors in check by these dynamic strategies. So I'm very positive here.
Okay, another one.
How do you choose which factors are all in your cancer model? Because the tumor microenvironment seems to be quite a big part of it. Sorry, I read it slightly differently, but that's what was the meaning, I think. Yeah, indeed, that's a good question, because all models are wrong and some of them are useful. So in the very early stages of cancer, we typically use also like, we include inside also the immune system, and we include other cells like stroma, etc. But in the advanced cancers, we typically see that if there is immune system, it's already fooled. And it also often believes already, so it's sort of cheated on by the cancer cells, that immune cells actually help the tumor to proliferate more. So in a way, we create a model which we believe is correct. Then we test it with data which come in. And if we see there is a big discrepancy, our model was wrong. So it's sort of reiterating process. It takes always a lot of time to actually come up with the right model, to have everything which is important in, but to make it still analytically tractable.
Okay, good. And there is one that appeared in my chat here from Terese, from one of the previous speakers, regarding the learning from errors part you talked about. Is this working through machine or human learning?
I'm not sure if I completely understand. Yeah, the errors. Yeah, the errors will be on both sides. So typically, of course. So in order to make right decisions, say for example in this kind of cancer treatment, you would want to have measurements all the time, and you would want to have accurate understanding of what's going on, and deciding all the time, right? So first of all, the data are sparse. They are often having a lot of errors. Maybe Veronica will talk about it in the next talk. So there is a problem already. How do we actually interpret this data? So this might be the machine part. And then our call, what's really going on, that's also often not completely correct. So often in these dynamic treatments, we have to see whether the tumor still grows or whether it decreases. And if you have really scarce measurements, you do not really know in which phase you are. And if the physician makes the wrong call there, what's really going on, then we will have a mistake in the response. So both.
Okay, good. Thank you so much, Katerina. Good luck with the rest of the research.
Thank you.
Okay, so let's move to one of your team members, actually. So Veronica Gasca, she's a master's student and at AFSE, and she will talk about matrix factorization to explore cancer cells. And you're related to Katerina's research team, no?
Yes, so I've just finished working on my master thesis, which was titled Applying Game Theory in Optimizing Cancer Treatment, and it was a data-driven approach. So Katerina was my supervisor on that, and today I'm not going to dive deeply into game theory part, as first we have only 5 minutes, and second, Katerina was already introducing it. But I will rather focus on data-driven part where we get some information that we can feed into theoretical models later on. So first of all, we have to think about cancer, like what we are dealing with. So basically, cancer samples are not only just tumor and that's it. They consist of many different types, cell types. And the issue about it is that because cancer samples will be so heterogeneous, then each of those cell types that is in this sample can react to the same treatment differently. So by applying, for example, chemotherapy, part of the tumor can be resistant to it, part can be sensitive. So within the same tumor, we can have whole spectrum of different reactions to the same treatment. So what is very important that we could identify the cell types, and we could track their response to the treatment such that we could adjust this treatment, the doses later on, to keep the tumor under control. So to do so, I was using matrix factorization, which is kind of simple concept. So we have the data in the form of a matrix. In our case, these are gene expressions data, where we have columns with different patients and rows with different gene sequences and the values that were measured. What we would like to find out is for each single patient, for each single gene sequence, what is the composition of the cell types in this tumor, and what are their proportions in there. So when we'll take a number, we can assume that there are 2 cell types, and we would like to find out what are the average expression of those genes in the cell types, and what are the proportions, such that after we will multiply these matrices, we will get back the measured values. So that's what I've been using. So basically, for my master thesis, I was working with 2 data sets. So my master thesis was basically about applying the this matrix factorization method to a big data set, such that we could extract those cell types from it. And then later on, we were also working with organoids data, where we had 3 patients and 2 different treatments applied to those, and the measurements in 3 different time points. So basically, what I was working on, those extracted cell types, I was tracking their proportions in the organoids to see the changes. So I found out that there are 3 main groups of cell types, which in this bigger data set, they had proportions that are depicted here, but that's not really relevant. When I was looking at those cell types in the organoids, in those 3 patients that I had measurements for, I could have noticed that for one of the patients, which will be better seen here, the proportions are really changing over the time, such that one of the cell types was decreasing in a proportion, other cell type was increasing, and thanks to this, I had my values such that I could adjust the model such that it will more match to the reality, since we get actual measurements. So since this moment on, I was using this information to work on the theoretical model. Which, as I said, I will skip in the presentation. So thank you.
Wow, thank you so much, Veronica, for this presentation. Let me see if there are any questions. And you did this for your master's thesis, right?
Yes, that was my master's thesis.
Wow. Congratulations.
Oh, thank you.
Okay, that's quite impressive. So let's see what questions will load. Okay, you mentioned the used RNA data. Did you use single cell data?
To be honest, I cannot say, because for me, there were RNA sequences, there were gene expression measurements, but from biological or side of this project, everything I was more like given. I was not really diving it into this information. So I think it's not single cell data, but I cannot say for 100%.
Another one. Have you tried applying this approach in imaging?
I was not working on imaging at all, because I wouldn't like to do my master thesis for 4 semesters or something. I had just one to do so, but I know that at my department there was one research project, and one of the groups was working on applying similar approach to imaging. However, I don't know how that was going.
Okay. Let me ask you something. I mean, since this is your master's thesis and it's already quite impressive, what will be your next work? What comes next?
Yeah, I'm hoping to go further with this research. So I'm hoping to start my PhD soon and continue on this topic.
Okay, good. I really hope that it goes well for you. I'm really impressed, to say. Okay, so thank you so much. Let's move then to our last presenter for today. It is Dr. Sangeeta Svabnastrita, and she's a postdoctoral researcher at MERL, which is at AFHML, if I'm not mistaken. So, and hi, Sangeeta. Hi. Your presentation will be about the computational modeling of the kidney.
Yeah.
Okay, so you have the floor. Please tell us more about it.
Inception. Is that?
You just have to, I guess, make it. Yeah, good. Yeah, we can see. I think that this is the first slide, no?
That's okay. So I would like to propose modeling as an. Initially, it was to be proposed as an alternative, but looking at all the work that everyone is doing, I would rather suggest it as more of a friend to experiment or a helpful aid to experiment. So I'm Sangeeta Svabnastrita. I work in CBITE in the group of Aurélie Collier here at Merlin. And I would like to introduce to you our research, which is computational modeling of diseases. So to begin with, the most popular example of modeling is the flight Boeing 777. It was designed entirely as a modeling by a modeling team. So starting from product proposal to detailed product design, even setting up where the factory would be, where to procure the raw materials from, where to test the product, the training system maintenance, everything was done via modeling, because these are expensive systems costing upwards of 100 million per item. So it's necessary to invoke modeling into this. And so necessary it is also to regenerative medicine, because it's not ethical and also very difficult to experiment and work with actual people data. So our idea is, can we bring this modeling to regenerative medicine? Focusing now on my research, I work with kidney. So the main function of kidney is to reabsorb water and salt that we put into our body, and then main functionality being removing urea and other toxins and also other drugs from the body. So when the kidney does not work, what happens is you have nausea, fatigue, weakness, confusion, high blood pressure, and many more tragic consequences. So what happens when people have acute kidney diseases or chronic kidney diseases? The first option for them is to have a kidney transplantation. If we look at the pie chart here, we see that the number of people requiring transplantation is actually a much higher percentage than the people actually getting transplantation. And to add to the worries, there is a major section of people who are getting added to this list every day. So our worries do not end here. But the one tiny ray of hope is that there is a bridging process here, where you don't get a kidney transplantation, but you can elongate the life of a person for some years, and that is dialysis. So it is a passive transport process. So what is done is you take the blood from the patient, then have some heparin shots to thin the blood out and pump it into a dialyzer. In this dialyzer, you use a solution which will clear the toxins out from the blood through membranes, and then the clear blood is then pumped back to the human. So this essentially mimics the role of a kidney, but it is a lifelong process, and once you are on dialysis, there is very slim chance that you will not be on dialysis, and it's a long process. So always researchers are working on how to improve this process, how to improve the toxin removal process in the membrane, and this can be done via modeling as well. So here I have a very simple representation of how the dialysis membrane would work. So we have our blood chamber, we have a membrane, and then we have our dialysate that we are seeing in blue. So what will happen is, in the blood chamber, the toxins will flow through, and then they would be removed to the dialysate via some transport equations. And here I have specifically shown what if we have a linear linear porous membrane, or if we have a wavy porous membrane. And wavy is something that is not tried, but theoretically we can show that it will have higher clearances. So this is something that the experiment people can then work on and try to improve. So via modeling, we can propose what kind of membranes you can use, we can test out different toxin levels. We can conduct multiple in silico trials without actually going to patients. We can test out flow conditions which might not be possible to try out in real patients. So all of these opportunities would be available to us if we start hooking up our experiments alongside modeling. So I hope I have convinced you a little bit about the benefits of actually doing modeling, because it is cost effective, it is time effective, and it helps us to personalize medicine and make those solutions available to patients quicker. So with that, I would like to finish, and yeah, I'm looking forward to questions and ideas.
Thank you so much for this topic, which is very relevant, huh? Let me see what questions will show up on the screen. Let's see, but let me first ask you, at which stage of your research are you? Have you completed it, or?
It's always continuing. It's never finished. So we keep working on things. Of course, our main aim is to have an actual instrument to propose to people. But yeah, before that, we need a lot of research. So we are in that stage, testing out different sensitivities, if it would actually be feasible.
So the ambition is to have a system to propose.
Yeah.
I understand. Okay, one question. Organ on a chip for organs. This is a trend. How do you see this working together with modeling?
Actually, a very good question. We do want to work with our research is aimed at informing these organ-on-chip models. So if you would like to see that work, that's also in progress right now, where we have proposed binding kinetics for certain toxins, and that would help us differentiate a little bit about how transporters work. It's getting into complicated territory. So, but yeah, we do work with organ on chip, and that's a very interesting avenue for us.
Okay. Oh, from Anna. Oh. What tools?
For modeling, we use COMSOL and VSEL. COMSOL is a paid service, but I think VSEL you can get for free. And both of these tools are amazing to work with. Actually, we are looking to bridge them some way so that we can use the data from VSEL in COMSOL.
Okay, let's see whether another question pops up. I think that's it. Okay. Okay, so I think that was it. Thank you so much, Sankita, for this lovely presentation. And I think also that our time is up for this session, so we must close this session. I would like to thank all who attended this event and for the lovely questions, and also, of course, to our speakers. It has been a great privilege for me to hear your groundbreaking research, and for me it was a great inspiration. So please note that the videos will be shortly available after the event on social media, and Pint of Science also will prepare the publication for the posters for those who had very good also proposals, but they will do so through posters. There will be an article discussing the event by the Moois Rijn Gazette. And I would like to ask also those who attend the event to please tune in for the next session that starts at 12 o'clock to the second link. And ladies, thank you all for joining us today to celebrate International Women's Day. And have a lovely week.
Thank you.
Bye-bye.
Thank you.
Bye.